Hello students, welcome back to my channel. In this video, I am going to give you MCQs from chapter 7, that is cell cycle and cell division. These questions are very much useful for your IP exam preparation. So let's see the questions one by one. Let's see the first question. Identify the phase that is present between two successive M phases. Options are telophase, anaphase, interphase, S phase. For this, the correct answer is interphase is the correct answer. Interphase is the correct answer. So, interphase is present between two successive cell divisions. So, cell cycle consists two phases, interphase and M phase. Interphase always occurs between two mitotic divisions. So, interphase is the resting phase that occurs in between two division phases, nothing but two mitotic phases. In between two mitotic phases, there is interphase. So, in the interphase, cell prepares for the cell division. So, let's see the second question. What is the duration of cell cycle in yeast? Options are 20 hours, 20 minutes, 90 minutes, 30 minutes. For this, the correct answer is 90 minutes is the correct answer. So, duration of cell cycle in yeast is about 90 minutes under optimal growth conditions. So, for bacteria, it is about 20 minutes. For humans, it is about 24 hours. Okay. So, let's see the next question. What happens in the S phase? Options are DNA synthesis and chromosome number remains same. DNA synthesis, chromosome number reduced. DNA synthesis does not occur, chromosome number doubled. DNA synthesis does not occur, chromosome number reduced to half. For this, the correct answer is in S phase, DNA synthesis occurs but chromosome number remains same. So that is the correct answer. Okay. DNA doubled in the S phase, that is nothing but DNA synthesis or replication occurs in the S phase. So each chromosome contains two sister chromatids. Okay, but the chromosome number remains same. Okay, so that is the correct answer. Let's see the next question. If a cell has 16 chromosomes in G1 phase, and how many chromosomes does the cell have in G2 phase? Options are 8, 16, 32, 64. For this, the correct answer is 16 is the correct answer. So, G1 is the gap phase. In G1 phase, chromosome number is 16. So, each chromosome contain one chromatid. After G1 phase, there is S phase. S is nothing but synthesis phase. In case of synthesis phase, DNA get doubled. So that's why each chromosome contains two chromatids. So chromosome number is still 16. Centromere not yet divided. So that's why chromosome number remains in the G2 phase also same. So the chromosome number of G2 phase is 16 only. Okay. Only extra chromatid synthesized in the S phase, but the chromosome number remains constant. Let's see the next question. Cells enter the quiescent phase, nothing but G0 phase, by exiting the cell cycle from which phase? Options are G1 phase, G2 phase, S phase, M phase. For this, the correct answer is G1 phase is the correct answer. Okay, G1 phase is the correct answer. Cell cycle has 4 phases. You know that G1, S, G2M. Okay, so in G1 phase, cell increases in size, synthesizes RNA and proteins, enzymes, and also it decides whether to divide or not. So the cell is deciding to divide or not. So it has decision point. G1 has a checkpoint. When conditions are favorable, it enters into the S phase. When conditions are unfavorable or cell is specialized to do a specific function, then cells exits from the cell cycle from G1 phase and then they enter into the G0 phase or quiescent phase. Okay, so the correct answer is G1 phase is the correct answer. So let's see the next question. So arrange the phases of karyokinesis in the correct order. Options are telophase, anaphase, metaphase, prophase. Metaphase, telophase, prophase, anaphase. Anaphase, metaphase, prophase, telophase. Prophase, metaphase, anaphase, telophase. So the correct answer is prophase, metaphase, anaphase and telophase is the correct answer. Let's see the next question. In which phase of cell division does centromere splitting occurs? Options are mitotic anaphase, meiotic anaphase 2, mitotic anaphase 1, meiotic anaphase 2, mitotic metaphase, meiotic metaphase 2, mitotic telophase, meiotic telophase 1. For this, the correct answer is mitotic anaphase 1 and meiotic anaphase 2 is the correct answer. So, during the mitotic anaphase 1 and meiotic anaphase 2, centromere splits and chromatids get separated. Those chromatids, they move to opposite poles. Okay, so the centromere division, centromere splitting occurs in mitotic anaphase 1 and meiotic anaphase 2 only. Okay, so let's see the next question. Chromosome compaction starts from. Options are leptotene, zygotene, pachytene, diplotene. For this, the correct answer is leptotene is the correct answer. Actually, in leptotene stage, the chromosomes get condensed. Okay, the compaction of chromosomes occurs throughout the leptotene stage. Okay, so during the first phase, that is nothing but leptotene of meiosis, the chromosomes they get condensed to form a chromosome structure. Otherwise, they are simply like thread-like structures, nothing but chromatin material. Okay, during the cell division only they become compact and forming a chromosome structure. Okay, let's see the next question. In which of the following phase bivalent formation occurs? Options are leptotene, zygotene, pachytene, diplotene. For this, the correct answer is zygotene is the correct answer. During the zygotene stage, the chromosomes start pairing together and this process of association is called as synapsis. Okay? Such paired chromosomes are called as homologous chromosomes. So, homologous chromosomes they started pairing during zygotene stage of meiosis. Let's see the next question. Chiasma or X-shaped structures appears in. Options are leptotene, zygotene, pachytene, diplotene. For this, the correct answer is diplotene is the correct answer. Actually, during pachytene, homologous chromosomes are completely paired. Crossing over occurs in between non-sister chromatids of homologous chromosomes. So, they overlapped each other. That is that results in the formation of X-shaped structures. Actually, they are formed in the Pakitine stage, but they are clearly visible later in diplotine stage. Okay, so they are formed in the Pakitine, but they are clearly visible during the diplotine stage. So that's why the correct answer is diplotine is the correct answer. Let's see the next question. Terminalization of chiasmata occurs in. Options are leptotene, zygotine, pakitine, diplotene. For this, the correct answer is diakinesis is the correct answer. After crossing over homologous chromosomes, they start to separate. The chiasmata begin to move towards the ends, nothing but terminal regions, okay, of the terminal regions of the chromosomes. So this movement of chiasmata towards the chromosome end is called as terminalization. So terminalization actually begins in the diplotene stage. So it is completed by diakinesis. Okay. So that's why the answer is diakinesis is the correct answer. So let's see the next question. So bivalent of meiosis 1 consists of. Options are 1 chromosome, 2 chromatids, 2 chromosomes, 2 chromatids, 2 chromosomes, 4 chromatids, 4 chromosomes, 4 chromatids. For this, the correct answer is 2 chromosomes, 4 chromatids is the correct answer. So, bivalent of meiosis 1 consists of 2 chromosomes, 2 homologous chromosomes, each one having 2 chromatids, overall 4 chromatids. They are present in a bivalent. Okay. Okay, students, these are the questions. See you in the next video. Thanks for watching.